In this video, we will be looking at the possible ways in which bouts of exercise could help to treat the effects of insulin resistance, particularly in muscle cells. First, let us recap on how insulin works. What is insulin and how does it work? Insulin is a peptide hormone that is produced in the beta cells in pancreatic islets in response to high blood glucose levels. Insulin helps to lower blood glucose by both inhibiting glucose production in the liver and promoting glucose utilization in our peripheral organs. How does insulin move glucose from the blood into the cells? When insulin binds to the insulin receptor at the cell surface, it triggers a series of reactions known as the insulin signaling pathway. The end goal of the pathway is to allow the glucose transporter, GLUT4, that is located in intracellular storage vesicles to be translocated to the plasma membrane. At the plasma membrane, GLUT4 can transport glucose from the blood into the cell. So, what is insulin resistance? Insulin resistance occurs when the body is still producing insulin, but the binding of insulin to the receptors does not trigger the insulin signaling pathway. This could be due to problems with the insulin receptors. As a result, there is reduced uptake of glucose from the blood into the cells. If untreated, this could lead to the development of type 2 diabetes. There are some possible ways in which exercise helps to counter the effects of insulin resistance. The first would be through the contraction-mediated pathway, which will be the main focus of this video. As we all know, we use many muscles during exercise. The continuous and repetitive muscle contraction during exercise acts as a stressor agent. During intense or prolonged exercise, activity causes depletion of ATP levels, which must be rapidly resynthesized. Enzyme adenylate kinase, or ADK, converts two ADP molecules into one ATP molecule and one AMP molecule. While the ATP molecule produced is used for energy, the AMP molecule attracts AMP-activated protein kinase, or AMPK. AMPK plays a similar role to AKT and phosphorylates the AS160 protein. AS160 is a RAP GTPase activating protein. When AS160 is not phosphorylated, AS160 maintains its target RAP in an inactive GDP-bound form, which retains GLUT4 in intracellular compartments. However, when AS160 is phosphorylated, the target RAP changes into an active GTP-bound form, allowing the translocation of GLUT4 to the plasma membrane. With more GLUT4 at the plasma membrane, there can be increased uptake of glucose from the blood into the cells.